Swayam Prabha. Digital India. Educated India.
Welcome back to the lecture series in animal physiology. So we are in week 2. So first 3 classes of the week 2 we have talked about the integumentary system. The next 2 classes of this week will concentrate on cartilage and bones. So before getting into the cartilage and the bone, let's ask a question to ourselves. Have we ever wondered this whole structure of our body with 2 limbs, 2 legs, the unique shape of the skull, these are all governed by a solid framework, and we call that as skull or skeleton sideways. Did it ever cross your mind that when possibly the skeleton actually developed? Like in the development phase, what I meant by that? We are conceived, a sperm and an egg fertilize, form a zygote, and then moves on. It forms an embryo, and then likewise, at what stage this framework takes a rigid structure? So, to tell you, by the 6th week of the development, like fertilized egg is formed, so the baby is conceived. 6 weeks down the line, approximately can say 1.5 months, the framework, what you call as a skeleton, is already formed. But it is not very rigid. Interestingly, at that stage, no bones were formed. Framework was there, but there was no bones. What does that mean? That means the initial framework which is formed in the mother's womb at the 6th week, by the 6th week I should say, is not the real bones. The framework is of cartilage. There was a framework. There's the whole shape of the whole, say for example, so let's to initiate with. So we are into week 2, and this is class 4, followed by will be the class 5. Okay. So today what we are dealing with is bone and cartilage. So where I was, so I told you that you have this framework, of bony structure is formed, but that was not really a bone. That framing network was at that stage, or is at that stage, is of cartilage. But then what is cartilage? So let me give you a few facts, and then we'll explore each one of these facts. In the mother's womb, by 6th week, I mentioned, so there are a few things which we'll highlight first. By post-fertilization, okay, where you have a sperm crossed with an egg and it fertilizes. By 6th week, skeletal framework, I'm just writing framework, okay. A skeletal or a skeleton framework, skeletal, or you can also call it skeleton framework, is already formed. So in other words, like this whole framework, it's in a I have drawn a cartoon. This framework is already formed, and this which are destined, these structures, structure of the skull, structure of the leg, and pelvic girdle and all these things, these were all destined to become bone. But initially they are destined, mark this word, they are all destined to become bone. But initially all these structures were cartilage. And there are two processes by which a bone is formed. One process is a cartilage is transformed into a bone. Okay, so those who all are non-vegetarian, those who have ate chicken or mutton or something, they must have seen a bone, or those who are even non-vegetarian, vegetarian, they must have seen a structure of the bone, right? You must have seen, or those who maintain dogs, the dog bone, you must have seen a model of the bone. So you'll see the structure is something like that, right? And at the top of it there is one small layer kind of a structure, which is kind of elastic kind of a structure out here, which you can see. That structure most of the time our parents or somebody will tell us that is a cartilage. Okay, that is indeed is a cartilage. But what is cartilage, will come to that. And this is the main bone. And those who are very good in anatomy or meat anatomy, they know that there is a canal. So this whole structure is hollow, and one calls it as the marrow area, which is also called the marrow or bone marrow. And there are two kinds of marrows which we'll be dealing later. One is red marrow, the other one is yellow marrow. We'll come later what does those words mean. But this is typically a bone, how it looks like. So one route for the formation of bone is from cartilage. So a cartilage becomes a bone. This is route 1, which is one of the most prominent route. And there is another route where there are some series of stem cells in our body which are called mesenchymal stem cell. In short, they write it as MSC. For you people, just to remember, this is called mesenchymal stem cell. And sometimes even there are fibroblasts which, given the local environment, transformed into bone. And it is not the whole cartilage of the body gets transformed into bone. Still there are cartilage wherever it is needed. We will come later into it. These are the two ways by which bone is formed in your body. Now, in order to understand the bone biology, which is also called ossification, OSSI, just remember this word, this formation of bone, this process is termed as ossification. In order to understand what is the genesis of the bone biology, you have to understand from where it is formed. So you have to understand what is cartilage. Once you understand what is cartilage, understanding bone will be very easy. Now, while I'm asking this question, what is cartilage, let me just recall a little bit of our first week when I mentioned that whenever and wherever it is needed, I will refer to those tissue level of organization. You remember there we talked about muscular tissue, nervous tissue, under the broad heading of connective tissues. Likewise, we talked about loose connective tissue, dense connective tissue. Okay, so the term connective tissue which includes blood and everything. There is another term which is called the loose connective tissue. And within the loose connective tissue falls these two words. So, if you have to make a mind map, so it is connective tissue, and within connective tissue you have loose connective tissue, and within loose connective tissue you have cartilage and bone. Okay. Right, and I mentioned you, it is from the cartilage you can make bone. This is the arrow, and this process is called ossification. Clear? Now, in order to answer what is the bone, we have to first of all answer what is cartilage. So, let's start with cartilage. How we define cartilage? So, this cartilage is basically the matrix of cartilage, the matrix of cartilage, and I have given you one example where cartilage is located. If you remember it, when I showed you the structure of the bone, I told you that in a mature bone, cartilage is located somewhere out here. And there is a term which is used for this. This is called articular cartilage. Articular cartilage. Okay. Fine. Now, the matrix of cartilage is a form of gel which contains polysaccharide derivative, polysaccharide derivative, and one such polysaccharide derivative is called chondroitin sulfate. Okay, and this chondroitin sulfate is one of the major component of the cartilaginous material. Chondroitin sulfate, represented by Ca, it forms kind of a complex with the protein in the ground substance. So, it forms a kind of a complex with protein thus forming term what we call as proteoglycans. And apart from this matrix, if one component of cartilage is matrix, there is another component which is the cellular component. And cellular component of cartilage is called chondrocytes. Now, when you see an immature bone, you will be surprised that this whole bone, you see this hatch line here, initially this whole bone is nothing but cartilaginous tissue. Initially, at the 6th week when the whole framework is formed, this is how it was. The whole thing was cartilage, okay, and there was nothing called articular cartilage or epiphyseal cartilage. We will come into that, okay. the whole thing was cartilage. The whole bone was nothing but a cartilaginous material. And there is a very interesting feature about cartilage. Cartilage cells, you see this cellular structure. So, what you see is the matrix, and within the matrix you are having the chondrocyte sitting here like this. Okay, this is how the cartilaginous material is formed. These circles are the chondrocytes. Fine. Now, there is an interesting feature about these chondrocytes. These chondrocytes secretes certain specific factors. These factors does not, so I am representing factor with F. These factor does not promote growth of blood vessels. And there is a technical term for this. This is also called secretion of anti-angiogenic factors. And there is a huge significance of these anti-angiogenic factors. Before I come to that, so by the 6th week, when this whole framework is formed, so these cartilaginous bones are not having any form of blood innervation. Information 1. Information 2, I mentioned here that they secrete certain factors called anti-angiogenic factors. Anti-angiogenic factors are the factor which does not allow the blood vessels to grow. So, this piece of information has immense significance in cancer therapy. Whenever there is a cancerous growth at XYZ location, those cancerous cells survives because they are continuously fed by the blood vessels. All the nutrients, all their debris are being removed. But think that these cancer tissues, if they are exposed to an anti-angiogenic factor, it will not allow the blood vessels to survive there. It will kind of destroy them. And in that situation, that cancer outgrowth which has formed will die out. In other words, if I had to give a diagrammatic explanation, say for example, this is the tissue and there is this cancer outgrowth which has taken place. Obviously, that has been supplied with blood vessels. These are the blood vessels which are supplying it. Now out here you put your factor, anti-angiogenic factor. I am representing by AAF. Okay. AAF. There are several anti-angiogenic factors. So, what this will do, this will kind of remove these blood vessels or kind of destroy these blood vessels, and eventually this tissue which is an outgrowth out here will be devoid of nutrients, and theoretically speaking it should die out. Okay. So, this is one of information which is very interesting about this particular cartilage material. Now, the physical properties of the cartilage depends on two factors. Factor 1, these are the physical properties. Okay. Factor 1, that is type and abundance of extracellular fiber. Type and abundance of extracellular fiber. Extracellular fibers, and there is second point, which is type and abundance of proteoglycans, and we have already discussed what are proteoglycans in our previous slide. Okay. Now the cartilaginous material, what you see, is surrounded by fibrous pericardium. Okay, it is surrounded by this is fibrous perichondrium, sorry, perichondrium, and which is the outer fibrous region of irregular connective tissue. Okay, this is that outer region of fibrous region of dense irregular connective tissue, and there is an inner cellular layer. This is that inner cellular layer, whereas this is outer fibrous region, fibrous irregular region. This is the one which provides the mechanical support and helps in anchoring with other tissues. Say for example, this is one surface, it has to anchor to another surface, helps in this anchoring or attachment. Whereas the cellular layer, the inner cellular layer, this helps in the growth and maintenance. Next, what we will talk about is the cartilage growth, how cartilage growth takes place. Okay, because from after this only we will have a fair idea how the bone is formed. Cartilage growth. So cartilage growth takes place in 2x2 process. One is called interstitial growth, interstitial growth. The other process is called appositional growth. Appositional growth. So both these are slightly different from each other. Say for example, you think of a confined space where you have a cartilaginous tissue. Okay, so let me draw it in 4 different stages. Okay, so imagine this single box is one confined space, and here you have a cartilaginous cell or a chondrocyte. Okay, so this is the matrix, and this is the chondrocyte, and this is the lacunae or the space where it is located. Now, one of the ways they grow is this particular, in that confined space, this particular chondrocytes starts to divide. While it is dividing, you are basically expanding the space. Okay, so it's dividing from single cell, it is becoming 2 cells. Then within this space, it creates a more lacunae or more empty space, and you have 2 cells like this. So what you are essentially doing is that you are expanding on all the directions, and while it does so, these two daughter cells which are formed, they secrete a newer and newer matrix in and around them. Okay, and thereby you are spreading out and you are growing. So as daughter cells secrete additional matrix, they pushed apart and expanding the cartilage form within similar. This is very similar to us. Say for example, an example of a balloon out here, and within one balloon you are inflating another balloon. So what this balloon will do, it is going to expand this one. So always remember it by the analogy of the balloon, right? So this is, if this is the first balloon B1, there is another balloon which is B2, which is expanding the balloon 1. So the next, this balloon 1 will expand a little further till it bursts out. This is one way by which it does so. And there is a second one which is called the appositional growth. Appositional growth is very interesting. In the appositional growth, what you are having is that you have these young chondrocytes in the lacunae, what we just now draw. Okay, so this is the lacunae where these young chondrocytes are present. Okay, and on top of it, you have another cell type which is the fibroblast. I am not putting any nucleus in them purposefully so that you make you understand. Okay, I am just shading it, but they do have a nucleus. Okay, and these are the dividing fibroblast. Okay. Now what happens, and this is a, okay, this is the young chondrocyte which is present there. Now these fibroblasts, these dividing fibroblasts in close proximity, and this is your perichondrium. You remember the perichondrium what we talked about in the previous slide? Okay, this is the perichondrium area, right? Now what happened, these dividing fibroblasts in close proximity, so they are dividing fibroblasts in close proximity with these chondrocytes, they get converted. So if I represent fibroblast as F, these fibroblasts get converted into chondrocytes. Okay, so fibroblast converted or differentiated into chondrocyte. So let's represent those chondrocytes at CF at the superscript, telling that they have come from the fibroblast. Okay, so this is another way by which they grow, and these fibroblasts then become chondrocytes and become part of this layer, and this results in the increase of the matrix. These matrix are started to spend, and what we eventually land up with is a new matrix formation. Okay, so with this new matrix formation, this whole cartilage layer keeps on expanding. So, and these older ones started coming down into the matrix. So that's how this matrix size eventually becomes much more bigger as it expands. So these are the two ways by which the cartilage material divide. So what all we covered today in this particular class, we talked about what is cartilage, what are the structure of the cartilage, and we talked about the two different growth mechanisms by which cartilage grows. And in the beginning of the class we talked about how from cartilage the bone is formed. So in the very next class what we will do, we will talk about the different type of cartilage and how the bone is formed. Thank you.